The human body consists of roughly 37 trillion cells, and their complex regulation and interplay manifests itself as a living, functional organism. At the core of life is the genome, whereby its integrity and expression patterns are, in part, determinants of the health of the resulting organism. This can be exemplified by diseases such as cancer, forming the topic of this video, in which cancer can be defined as a group of diseases generally characterized by genomic instability and uncontrolled cell division, leading to the invasion of surrounding tissues and eventual migration to distant sites. Specifically, I shall be focusing on estrogen-receptor alpha, or ERA, positive breast cancers, which represent around 75% of all breast cancer cases. Estrogen and its subsequent activation of ERA, which functions as a transcription factor, has a multitude of physiological roles in primarily females via the expression of ERA target genes. Of particular interest is the role of ERA in promoting the proliferation of cells in the breast and uterus, which can become dysregulated and exploited by ERA-positive breast cancers to support tumour growth, as some ERA target genes include growth factors, but also other mitogenic genes. For this reason, the first line of treatment for ERA-positive breast cancers is anti-hormone therapy, a classical example being the use of the drug tamoxifen, which is an ERA antagonist, effectively mimicking the estrogen molecule in design. Tamoxifen treatment can result in the inactivation of ERA. However, via several proposed mechanisms, it can also result in tamoxifen-induced apoptosis. This creates a selection pressure for which the cancer must evolve. One way to deal with this is via ERA downregulation, thus conferring tamoxifen resistance to avoid apoptosis. However, by downregulating ERA target genes will also be downregulated, resulting in the loss of ERA-related proliferation and growth, creating a new problem for the cancer to deal with. One potential solution may be via the role of hypoxia, whereby more than 50% of solid breast tumours have a hypoxic microenvironment, hypoxia being a state of low oxygen concentration at the tissue level. It's possible that hypoxia can downregulate ERA expression, but through the application of tamoxifen treatment, which is effectively replicated for a lack of estrogen, remaining ERA is inactivated. Interestingly, it seems that hypoxia can simultaneously generate tamoxifen resistance, in which the cancer no longer responds to tamoxifen, whilst regulating a fraction of ERA target genes, whereby it was previously thought these were mutually exclusive due to ERA inactivation. First, researchers performed a Western blot experiment to determine ERA expression under hypoxic conditions for 1 month, either with estrogen, labelled as E2, or without estrogen, which was replaced with ethanol, so a non-compatible ligand of the ERA, labelled as ETOH. A significant decrease in the expression of ERA was observed under hypoxic conditions, which is cobalt chloride, relative to the control, which are normoxic conditions, as shown in the purple and yellow boxes, thus confirming that hypoxia can trigger ERA downregulation. These researchers using MCF7 cells, which is a cell line from an ER-positive breast cancer. Using microarray experiments, it was discovered that under estrogen treatment, 548 genes were expressed in normoxic cells versus 311 under hypoxic conditions. However, more than half of these will display a loss for estrogen and thus ERA-dependent regulation under hypoxia. This differential expression can be visualized in the figure, where red indicates expression above baseline whilst blue indicates expression below baseline. Interestingly, high degrees of variation can be observed across the ethanol samples under both control versus hypoxic conditions, thus suggesting hypoxia-specific regulation of ERA target genes. Furthermore, variation across the ethanol versus estrogen samples under hypoxia indicates ERA target gene expression can occur independently of estrogen availability. To investigate how this could be occurring, chip experiments were performed targeting histone-free lysine-27 acetylation, a histone post-translational modification indicative of active enhancers, and interestingly it revealed a particular enrichment at a key subset of ERA target genes, some of which are known to promote malignant tumour progression, for example AREG, a growth factor capable of promoting proliferation. The upregulation under hypoxic conditions was confirmed experimentally, as shown in the figure, where estrogen independent, so the white boxes, expression of AREG is significantly upregulated in hypoxia relative to control. However, how is it that these key genes are specifically upregulated under hypoxic conditions? The answer may lie in the role of HIF-alpha factors, these being the key transcription factors that mediate the cellular changes associated with hypoxia. this being explored in ChIP-seq experiments for HIF1-alpha and HIF2-alpha binding, which revealed over 80 ERA target genes showing binding affinity for HIF-alpha factors. Interestingly, binding was enriched to several of the same genes that had been reportedly acetylated under hypoxia, suggesting that HIF-alpha factors are responsible for this, and potentially the activation of these genes, or at least their priming. This being exemplified by the estrogen-independent expression patterns of AREG under hypoxic conditions as shown in the maroon bar, whereby a roughly 5-fold reduction in expression occurs when HIF1-alpha is knocked down by RNAi relative to the control. To validate that hypoxia can permit the expression of some ERA target genes, whilst under tamoxifen treatment, reverse transcription quantitative PCR was performed to rapidly assess gene expression change. Using the example of AREG again, we can observe the conservation of estrogen-independent mRNA expression when exposed to just hypoxic conditions or hypoxic plus tamoxifen conditions. Thus, hypoxia can save the expression of certain ERA target genes during tamoxifen treatment. Due to the expression of genes such as AREG which are expressed in a tamoxifen-resistant manner, they could potentially act as predictive markers for anti-hormone response, helping to inform clinicians that a change in treatment is required. Furthermore, the data collected suggests some genes are upregulated in an estrogen-independent manner in hypoxia due to the role of histone acetylation. Thus, histone-free lysine 27 acetyltransferase inhibitors could be a potential candidate for the treatment of tamoxifen-resistant ERA-positive breast cancers. Further research is required to elucidate what transcription factor or factors are responsible for the tamoxifen-resistant expression of the 150 ERA target genes during hypoxia. Could it be mediated directly by HIF-A factors, which have shown affinity for 80 ERA genes? Could it be via the indirect action of HIF-A factors that modulate the expression of other transcription factors that can then regulate ERA genes? Could it possibly be ERA itself, although this does seem incredibly unlikely considering its downregulation as well as its probable inactivity due to tamoxifen treatment. This therefore requires further experimental probing. Maybe you could narrow potential candidates down via ChIP experiments against the 150 ERA target genes and then validate gene expression through the use of a cell-free expression system. Furthermore, the experimental validation of the acetylation capabilities of HIF-A factors may also be interesting, as currently the relationship is just correlated. Potentially this could be achieved via in vitro acetylation experiments, for example using radio-labeled carbon-14 acetyl-CoA.